So good morning students. Today we are going to see the regulation of acid-base balance by kidney. See, if the acids are excess in our body, the kidney will eliminate the acid by excreting the acidic urine. If the base is excess in our body, the kidney will eliminate the base by excreting an alkali urine. Okay. So in our body we have two types of acid. One is volatile acid, other one is an involatile acid. The volatile acid is carbonic acid, which will be excreted or which will be removed effectively by the lungs, because lungs excrete or exhale carbon dioxide. So as carbon dioxide is part of the carbonic acid, it will be expelled. The involatile acids, these acids are the products from the protein, the dietary protein, and these are to be removed, and this was done only by the kidney. So kidneys are the powerful regulators of the involatile acids. About 80 milliequivalents of involatile acids to be eliminated from the body. Kidney regulate the H plus ion level in the blood, okay, H plus ion level in the ECF by two important mechanisms. One is H plus ion secretion and bicarbonate reabsorption. Other one is it forms new bicarbonate. It's a very important event taking place in the kidney only. So now we'll see how H plus ion secretion and reabsorption of bicarbonate occurs. So H plus ion secretion and bicarbonate reabsorption. Bicarbonate is the filtered bicarbonate. So the filtered bicarbonate is reabsorbed. So say this is the peritubular capillary which is surrounding the nephron or the tubules, and this is the tubular epithelium. T stands for tubular epithelium, and this is a tubular lumen, and the lumen which contains the tubular fluid. Okay. Now what happens? The H plus ion secretion is done by an exchanger, H plus Na exchanger, or Na plus H plus exchanger. So this exchanger is an example of a secondary active transport, and this was operated after the primary transport occurring at the basal membrane. So the primary transport is sodium, potassium, ATPase pump. So when this pump is operating, sodium is pumped out. So the level of sodium is low in the tubular epithelial cell, and this causes sodium to enter into the cell for exchange H plus ion, and this is done by the sodium hydrogen exchanger. Now, where comes the H plus ion is coming? So it is coming from the metabolism of the cell, H2O or CO2, or it will be the CO2, it will come from the blood also, it will be diffused from the blood also. Now this H2O and CO2 forms H2CO3, which dissociate in the presence of the enzyme carbonic anhydrase into H plus and bicarbonate. Okay. So this bicarbonate is exchanged to the blood for chloride ion. So this is a mechanism taking place inside the tubular epithelium. H2O and CO2 forms carbonic acid, and this carbonic acid dissociates into H plus and bicarbonate. And all these reactions taking place in the presence of the enzyme carbonic anhydrase. Now this H plus ion is secreted into the lumen, and this H plus ion combines with the bicarbonate which is filtered and forms H2CO3, which dissociates into H2O and CO2. Again, the CO2 will be diffusing into the epithelium. So this way the H plus ion is secreted and bicarbonate is reabsorbed. So the point to be noted is the filtered bicarbonate is not the bicarbonate which is reabsorbed. Okay, this bicarbonate is not the same as this bicarbonate. Anyhow, for one H plus ion which is secreted, one bicarbonate is reabsorbed. Second point, this was done by the secondary active transport, sodium hydrogen exchanger. So this is a mechanism taking place in the proximal tubules, collecting in distal tubules, early part of distal tubules, and thick ascending limb. So this is the mechanism taking place in these segments. In collecting that, there is a different mechanism how H plus ion is secreted. So in collecting that, it is the same picture, but except for the presence of a pump, that is H plus ATPase pump. Here there is no secondary active transport as we saw in the other parts of the nephron. In collecting that, there is a cell. The name of the cell is intercalated cells or I cells, and they contain the pump H plus ATPase pump. It is a primary active transport, and this pumps out H plus ion. Okay, so this H plus ion is coming from the same carbonic acid, that is from the H2O and CO2, and this H plus ion dissociated, the H2CO3 dissociated to H plus and bicarbonate is exchanged for chloride. The same process. Only thing is here you have a specialized pump called H plus ATPase pump, and the points to be added is this is a primary active transport, unlike the secondary transport which is taking place in other parts. Second, this pump is activated by aldosterone. So aldosterone is a hormone that will activate this pump, and it is a hormonal effect, a stimulus on this pump. Now, in the previous two slides, what we saw is H plus ion is secreted and bicarbonate is reabsorbed. Now here, the secreted H plus ion is around 4320 milliequivalents of H plus ion is secreted, and for reabsorbing 4320 milliequivalents of bicarbonate, which is the filtered bicarbonate. Okay, so this much H plus ion is utilized for the reabsorption of bicarbonate. And we know there is an 80 milliequivalent of H plus ion to be excreted, that is from the non-volatile acids. and this was done by the other substances. So we don't have the bicarbonate buffering. Bicarbonate is not helping here, so we have the renal buffers that is going to help in the secretion of this much H plus ion, 80 millicoulombs of H plus ion. So these renal buffers are the phosphate buffer and the ammonium buffer. Ammonium buffer. Okay, so in this process, buffering by the renal buffers, the new bicarbonate is formed. As I told you, there are two ways. One is secretion of H plus ion and reabsorption of bicarbonate. Other one is formation of new bicarbonate. So formation of new bicarbonate takes place by the renal buffering action. The role of the renal buffers in PCT, if you see the same parts: peritubular capillary, tubular epithelium, and tubular fluid or the lumen. And here, what happens? The phosphate which is buffered, HP4 2-, a dibasic phosphate which is filtered, it is in the lumen, and this phosphate is combined with the H plus ion. So H plus ion is buffered by this phosphate and it forms H2PO4. Okay, H2PO4. So this H plus ion is buffered with this phosphate. Okay, so usually this will always come with this salt. Okay, sodium, Na plus HPO4. Now this H plus will combine with the phosphate, and the sodium will be exchanged. Okay, the sodium is exchanged by the same sodium hydrogen exchanger. As you saw in the PCT and other parts, we have the sodium hydrogen exchanger that is going to do this action, and this H2PO4 is eliminated in the urine. So where comes the H plus is coming? It is coming from the H2O and CO2, the metabolism, or from the carbon dioxide coming from the blood, forming the same reaction H2CO3, which is associated into H plus and bicarbonate. Now this bicarbonate is termed as new bicarbonate. So this new bicarbonate is transported to the blood for exchange chloride. So this is the mechanism how the phosphate buffer acts. Now comes the role of ammonium buffer. So ammonium is derived from the amino acid glutamine. Okay, so glutamine is produced in the liver and it comes in the blood and it enters the tubular epithelium, and the glutamine is also filtered. And this glutamine is diffusible. It will enter into the tubular epithelium and it is metabolized into 2-bicarbonate and 2-ammonium. So this 2-ammonium will exchange for sodium by an exchanger, sodium-ammonium exchanger. So this ammonium, which is pumped into the tubular fluid, combines with Cl- and forms NH4Cl-. Okay, NH4Cl-, and it is eliminated or excreted. Okay, now this 2-bicarbonate, which is termed as new bicarbonate, and that will enter into the blood for exchange chloride. Okay, so this is the ammonium buffer. So this ammonium buffer is more powerful than the phosphate buffer. Phosphate buffer usually removes about 30 to 40 millicoulombs of H plus ion only, and the remaining H plus ion, say about 50 millicoulombs, will be eliminated by the ammonium buffer. So these renal buffers, they act after the urine reaches the limiting pH of 4.5. So what happens in H plus ion secretion and bicarbonate reabsorption? The urine pH reaches a maximum of 4.5. It is the limiting pH. Okay, so after that we have to eliminate the H plus ion. That was done by the renal buffer. The first is the phosphate buffer and second is the ammonium buffer. And we have the another buffer which is acting in the collecting duct especially, which is the ammonia buffer. Now this ammonia, which is filtered, okay, it is filtered ammonia, and when it comes to the collecting duct, I told you in collecting duct you have a pump, that is H plus ATPase pump, and this pumps H plus ion, and this H plus ion combines with bicarbonate ammonia and it forms ammonium. Okay, now this ammonium is not diffusible through the membrane or the luminal membrane in the collecting duct, so it is eliminated in the urine. So where comes H plus ion is coming? The same mechanism, H2O and CO2 forms H2CO3, which is associated to H plus and bicarbonate. Okay, this bicarbonate is the new bicarbonate. Okay, for exchange chloride ion. So this is the way that ammonia acts as a buffer. The filtered ammonia combines with H plus ion and it is eliminated as ammonia. This is the role of the ammonia buffer. So to sum up, the H plus ion in ECF is regulated by either by H plus ion secretion and reabsorption of filtered bicarbonate. Okay. Second, forming new bicarbonate. Here comes the role of the renal buffers, phosphate and ammonium buffer. So they are the very important buffers that is eliminating the excess 80 millicoulombs of H plus ion, which is synthesized by the non-volatile acids. And this H plus ion secretion and reabsorption bicarbonate is like a mutual. So H plus ion is secreted, also the bicarbonate or the base is added so that it maintains the H plus ion. When H plus ion is very high, the secretion will be reduced and the reabsorption will be. So when there is H plus ion is very high, the secretion will be increased and reabsorption will be more. When the H plus ion level is low, okay, say in alkalosis, the secretion will be reduced and there won't be any reabsorption. Bicarbonate will be excreted in the urine. So this is the way the H plus ion is handled by the kidney. Thank you.